Let's invite our next, let me see, our next speaker. Our next speaker is Professor Khong Heng from University Malaya, Faculty of Applied Science, University Technology Mara, Cawangan, Sarawak. He will present or he will share his research. I think it's considered the group research entitled Antimicrobial, Antioxidant and Anticancer Potential of Banana, Musa paradisiaca, Sekaki, Florets from Sarawak. So let us invite Professor Khong. The floor is yours. You have 15 minutes. The bell will ring at 12 and 15 minutes and another 5 minutes for Q&A. All right.
Thank you, Chair. Actually, I already informed the person in charge that just play the video.
Yeah, technical.
In order to keep the time. Otherwise, I will exceed the time.
All right, I will play the video now.
Thank you.
Thank you, Mr. MC.
Good morning, everyone. Welcome to this session, Banana Biomaterials and Bioproducts. Thank you for your presence. First of all, I would like to take this opportunity to thank the conference chair, Prof. Yasmin Osman, and the organizer, University Malaya Center for Research in Biotechnology for Agriculture, extended an invitation to me and providing me an opportunity to share my work related to the banana. My presentation outline are including the introduction, methodology, findings, conclusion, and recommendation. From banana peels, we have extended our work to banana blossom. Therefore, this morning, I'm delighted to be here to share with you about our current work on the banana blossom, which entitled Antimicrobial, Antioxidant, and Anticancer Potential of Banana Floret from Sarawak. It is worth to note that the nutritional and the medicinal property of Musa species are not only limited to the fruits, but also other parts such as peels, roots, stem, leaves, and blossom. The banana blossoms, for instance, have long been used traditionally to treat diabetes and asthma, reducing the risk of excessive bleeding, as well as to relieving malnutrition in infants and diarrhea. The banana leaves, on the other hand, can be used as wound, and while the ash obtaining from the burning of the leaf is commonly used to alleviate asthma and wheezing. The immense medicinal benefit from the blossom is greatly contributed by its nature of having a high content of unsaturated fatty acid, dietary fibers, vitamins, and proteins. Previous findings also indicated that blossom is a good source of beneficial nutrition for human diet, similarly to other fruits and vegetables. Let's now turn to the previous study on Musa species. In recent years, research that have been undertaken revealed that the Musa species blossoms serve as natural sources of valuable phytochemicals, especially flavonoids and terpenoids, which are known to exhibit potent oxidative properties. Although humans are equipped with the built-in antioxidant mechanism against cancer-causing free radicals damage, it seems to be beneficial to obtain extra antioxidant in the diet, especially from the fruit and vegetable, as we understand that the present oxidant will help in increasing the resistance towards disease-causing cellular damages. Previous pharmacological study on the blossom of Musa species also demonstrated that they possess antimicrobial activity against Staphylococcus and Pseudomonas. Other significant pharmacological properties of blossom is that the blossom extract were also found to show cytotoxic and anti-proliferative activities against several cancer cells. For instance, human colon cancer cell HT29 and HCT116, as well as HeLa and breast cancer cell. In addition, there are several reported studies on the discoveries of valuable phytochemicals, such as coumarin, flavonoid, tannins, phenol, steroid, and triterpenes, from various parts of Musa species cultivars. Despite his ethnomedicinal uses of the blossom, the lack of exposure and knowledge limit the application of the blossom to be used only as organic fertilizer in the plantations. Previous study demonstrated that blossom is a natural source of antioxidants. However, there is no recorded study on the chemical and biological evolution of the Musa species cultivar in Sarawak. Hence, this study was aimed to determine the total phenolic content, total flavonoid content, antioxidant capacity, and antimicrobial property of different polarity solvent extract of Musa paradisiaca blossom, as well as to screen the anti-cancer property of the extract against the human prostate cancer, DU145 cell lines, and human cervical cancer, HeLa cell line, based on the MTT assay. The flesh blossom of the Musa paradisiaca was collected from the banana tree Sendirian Bahar, Kuching, Sarawak. The study materials Musa paradisiaca is locally known as Pisang Sekaki. It is a hybrid of between the Musa acuminata and Musa babesioana. The blossom was first separated into the brake part and the floret part. The floret were then cleaned and air-dried under adequate ventilation for 8 days at room temperature. The dry floret were ground into a fine powder using an electrical cutting mill. The ground sample was later stored in a vacuum storage bag prior to further processing. The dry fine powder floret samples were macerated in hexane, chloroform, ethyl acetate, and methanol to yield the hexane, chloroform, ethyl acetate, and methanol crude extract respectively. The prepared crude extract was then to determine the total phenolic content, total flavonoid content, and antioxidants activities. Besides, the microbial activity were assessed by the MIC and MBC assay, while the cytotoxicity was evaluated in vitro based on the MTT assay. The total phenolic content and the total flavonoid content of different extracts of Musa paradisiaca floret are summarized in this table. The TPC for the chloroform extract and the ethyl acetate extract were higher compared to the TPC of methanol and hexane extract. The TPC result correlated with the study on the Musa paradisiaca blossom, which demonstrated that ethyl acetate fraction recorded the highest phenolic content compared with the other solvent fraction. The TPC recorded in this study was also higher compared to that in the Musa paradisiaca cultivar P. rostari variety from another Malaysia region. The trend observed in the TFC was different compared to that in TPC, in which the TFC of the extract increased with the increase of the polarity of the solvent used. However, the flavonoid content obtained was relatively higher as compared to the values recorded in other Musa paradisiaca flower variety from Kuala Terengganu. Our findings are in agreement with other previous studies on other plants, which signify that the extraction of flavonoid is improved in solvent with a higher polarity. Among all the abstracts, the maximum antioxidant potential was observed in ethyl acetate, followed by chloroform, methanol, and it was the lowest in hexane extract. In comparison, the variety of these IC50 values signify that the antioxidants potential of banana floret is dependent on the region of cultivation as well as the cultivar of Musa species. On the other hand, the antioxidative property in these studies show a linear correlation with the phenolic content in the abstract, which increasing phenolic content contribute to better antioxidant capacity. Based on the MIC results, all the abstract display an active inhibitory effect towards all the tested pathogens. The extract were highly potent against E. coli, with the MIC values comparable to that of streptomycin sulfate, followed by the Pseudomonas aeruginosa. The antibacterial properties of the abstracts were further extended to MBC assay, and the data obtained correlated with the MIC assay in which the strongest inhibitions were observed in E. coli. This depicts that the Musa paradisiaca floret could be utilized to suppress the foodborne illnesses such as diarrhea and is a natural alternative to commercially available chemical preservative used in food products. The cytotoxicity of the floret extracts on DU145 and HeLa cell was a time-dependent inhibitory effect. At 24 hours, the cytotoxicity on DU145 and HeLa cancer cell line was found only in the chloroform extract. The activity was lower than the positive control. At 48 hours, the ethyl acetate extract showed the highest cytotoxicity on the DU145 cell, which was comparable to anti-cancer drugs 5-fluorouracil, whereas the chloroform extract showed the highest cytotoxicity on HeLa cell, which were lower than the anti-cancer drugs. However, the methanol extract showed no cytotoxicity on both cancer cells. In addition, the ethyl acetate extract demonstrated the highest selectivity index SI of 21.10 on DU145 cancer cell at 48 hours, which was superior to the ciprastine and 5-fluorouracil. Furthermore, all abstracts at the concentration below 100 microgram per ml exhibited no cytotoxic on the human dermal fibroblasts for 24 hours and 48 hours. The ethyl acetate and the chloroform extract at the concentration of 1000 microgram per ml for 24 and 48 hours show cytotoxicity on the human dermal fibroblasts, since they gave less than 80% of the cell viability when compared with the control. Likewise, the anti-cancer drugs at the lower concentration show higher cytotoxicity on the human dermal fibroblasts. In conclusion, the findings from these studies are evidence that Musa paradisiaca, cultivars Sekaki, floret exhibit high antioxidant and strong antimicrobial potential, as well as the moderate cytotoxicity against the DU145 human prostate cancer cell line. Additionally, the higher phenolic and flavonoid content are supported by the higher antioxidant property as compared to the cultivars used in the previous study. The floret, which are locally consumed functional food, could be a potential antioxidant agent and could be used as the medicament against the illnesses associated with free radical damages. Further studies are suggested to explore the vast medicinal values by assessing the in vivo biological activity as well as identify the phytochemical constituents to validate their pharmacological activities.
Thank you, Prof. That was very interesting and
For
very
your
trust.
information, the detailed work has been published in
it's okay, I think can stop here,
Scopus.
yeah.
Okay. Yeah, I was quite like, how come I see Prof on the line and then suddenly you're still talking? So it's actually the video.
Video, yeah, there's a video,
It's
yes.
efficient and effective in a way that you can keep your time and everything else. And
Yeah, because originally it's already exceeded, so I try to cut the intro, so to keep the time. Yeah, sorry for exceeding a little bit. Yeah.
No, it's fine. Okay, I'm going to open the floor for question for Prof. Khong, especially on her on the group finding of this antimicrobial, antioxidant, and anti-cancer potential, for the flowers of the banana. So I remembered my mom always, we love to eat the jantung pisang, so we never knew. Then we always throw away the flowers, but my mom did actually say you should eat that also because it has some, she said it's good for definitely everything. She would say that anti-aging, so all of us were like, so
in fact, in
sorry?
fact the floret part is higher biological activity compared to the bread part.
So this is only for pisang Sekaki. What about other form of pisang? Have you looked at it?
Because in my area, actually the company have planted more than, I think, about 200 acres. 100 acres is a contract farm. He owned about another 100 acres farm, and currently he is the banana chips manufacturers. So in one of the occasion, actually, I'm trying to introduce my cream, actually derived from the banana extract. But then he asked me to solve his waste problem because he had tons of banana peels at the factory there. So he doesn't know how to dispose it or how to turn into dollar and cent. So that is before I venture into this research. Actually, I started with the gluten-free flour derived from the banana peels. So
banana peels, not the florets lah.
Initially, that is how I started venture into research on banana. So initially, I started with the banana peels. I turned the banana peels into flour, and because it is very high nutrition, in fact, the percentage of the protein content is double compared to the fresh of the banana. So so it's attract us, and due to the nutrition value and also the gluten-free content, so we have converted into gluten-free flour. And currently, actually, he do manufacture in a small scale to meet the local market first. In fact, he got a how to say, a business from China, but then he doesn't cannot meet the demands. So actually, in fact, the company trying to look for financial support from the state government, from the central government, in order to extend his production line.
So this so making banana powder, banana flour to make, you know, anything. And
yeah, that is the initial
high protein.
Yeah. And after that, as we move on, and we found out that this Pisang Jantong, or we call blossom, is also one of the potential anti-cancer agent. Probably we have encountered, come across a few published paper on this. In fact, I think in Taiwan they have marketed it as one of the either drugs or herbal product. They also claim that there is anti-prostate cancer property. That is how lead us to explore to this research further. In fact, we also noticed that it is contain very high antimicrobial property, and it serves as a natural preservative. And we have proven in the edible bird net product, as well as the, in Sarawak, we are very well known with the cake lapis. And cake lapis manufacturer, actually, they have this problem because the cake lapis cannot last long. The most is for few days without keeping in the freezer. So we also have tested at our laboratory, and in fact, he really helped to preserve the cake or biscuit in a much longer period. In fact, the cookies that we make, I think it lasts for 2 years, also still in a good condition, without any preservative. Yeah.
Great, Prof. Khong. This is something very interesting that we can look at more towards looking at the waste product and to cycle it into something functional and something something useful. Thank you for your presentation.
Yeah,
it was
thank you.
A really good one. As you can see, you have Prof. Khong corresponding email. So for the floors, anybody who would like to collaborate or ask more, to contact Prof. Khong for further discussion.
Yeah, we welcome you. Yeah, in fact, we also have a conference coming, end of this year. So you can browse more information at UiTM Sarawak official website. Yeah, thank you so much for your kind attention.
Yeah, thank you, Prof. Khong. Okay.